Our last presentation for day one comes from Nystrom, CTO at Peptilogics. He's going to be talking about AI drug discovery training. Nick, how are you?
Hey, Brian. Happy to speak with you.
Yeah, great to have you again.
Okay, so thanks, everybody. I'm here today to talk to you about a new role I have. I'm with Peptilogics now. I'm CTO there. And I'd like to talk about AI-driven peptide drug design. This, to me, I was asked to speak in this visionary perspective track. And to me, this is what HPC and AI is all about, because this is solving a problem of pressing need for us as a society. And it's one for which traditional means are coming to a halt and becoming more expensive, more time consuming. So I'd like to tell you about what the challenges are, some of the progress we have made on training these sorts of models. So first, just legal disclaimers for the record. Let's start with, why is this a problem? Looking at the time to a drug approval, the average is now 10.5 years from phase 1, which means safety, to regulatory approval. That's a long time. The likelihood of getting from phase 1 to completion is only 7.9% on average. Certain things are higher, maybe as high as 20, but they're still low. And the cost is extremely high, and it's increasing. So the bar graph at the bottom of this page is looking at disease areas where there are at least 5 data points. And this is from 2 well-regarded studies. The first is the top bar and the lower 3. These are by disease area. And so we have here our cancer drugs on the top, and they're as high as $4.5 billion per drug. And then going down, a very widely cited study from 2016. I'm sorry about that. From 2016, the average across the board is 2.8 billion, and that incorporates some proprietary data. So it's a fair estimate. The bottom line is we need to do better. Well, one way to do that is to look at new modality. Existing modalities, small molecules are the norm. And by that, we mean molecules of about 900 molecular weight or less, and typically not any sort of protein or peptide structure, so not amino acid based. Biologics and multiclonal antibodies are on the rise. That's the blue curve. And these are doing well, but they can be very expensive to produce. They can be very complicated in trials. What we're looking at is peptides. Peptides are a class of small proteins. And I'll show you a bit more about what that means for those who haven't seen this. And they have a number of advantages. One is a manufacturing scale up. They can be incorporated into oral formulations to take easily. We can get them into cells for intracellular targets, and so on. They can treat what we call undruggable targets, which are things like protein-protein interactions, and some of those intracellular spots that small molecules have a very hard time getting to. And so that means that these have a very high growth opportunity. And these are essentially an untapped area that has been neglected compared to other drugs. Despite that, when they work really well. And we see that on this grid of brand names on the left. So when a peptide drug is developed, and they're not that common, they tend to be blockbusters. They do well. And the reasons for that are on the right. So the middle column is peptides, plus or where they have an advantage, plus minus is strengths and weaknesses. And so what we see is they're the only real modality here between small molecules, biologics, and peptides, where we have a plus in all of the rows. And the real strengths here are that because these are essentially nature's signaling molecules, they're what bind to proteins, they can be very specific. And by that, I mean they can bind to the target we want in a certain cell that causes disease, but not to other molecular targets that would cause side effects. And that means they don't have drug-drug interactions. That makes a lot of the clinical trials potentially higher success. And so that's what we're looking at. Where we are now, Peptilogics was founded in 2013, but very recently we started building this platform. I joined the company in January of 2021. And in the past year, we have made significant progress building this platform for AI-driven drug design. Our goal is to have the world's first end-to-end peptide drug design. By that we mean AI prediction of compounds, synthesis, and assays all the way through. So for a bit more of what that means is our platform is folding in proprietary compute technologies and algorithms. Scaling those up, we've built our own supercomputer. I'll say a few words about what that looks like. We've synthesized our own peptides on our custom designed synthesizer. And the point of that is to obtain a lot more data more quickly. As we just heard in the previous talk, data is the key to this. And in this field, with peptide and protein science, high quality data sets are rare. There's a lot of data. It tends to be a very mixed quality. And from source to source, it is very inconsistent. So what we're pushing forward with is a solution to be able to do this at scale. And rather than the long timeframes we showed a few slides ago, 10.5 years and billions of dollars, to make that scalable. So we can look at arbitrary targets, develop peptide hits, leads, and drugs much more efficiently. On the right is a quote from Andy Hawk, who many of you know, VP of Product at Cerebras. And there's someone we've been working with since last summer to look at bringing training times down to interactive timescales to really do hypothesis testing and drive the science. The beauty of peptides is they can be engineered. Because they are composed of amino acids, essentially in a building block sense, we can design them. This is an antimicrobial peptide. This was the first one that was formed by Jonathan Steckbeck, our CEO and founder. They got the company going. On the left is a natural peptide. And the point there was that this had potentially high antimicrobial properties, so anti-infective, but it was toxic. It had very strong inflammatory properties. Jonathan's hypothesis was he could design out those negative inflammatory properties and design in improved activity, better stability in our system. And that's what happened. It turned out to be a very highly potent compound. It is now in phase 1B trials. We expect to be enrolling our first patients imminently. And rather than the half-life of normal peptides, which we metabolize with proteases within a couple minutes, this one, our lead compound 206, has a 15-hour plasma half-life. And so that means it stays in our body long enough to do everything it has to do. And so that was good. But it was a typical human design process. It took years. And that's where we wanted to do better. So we scaled out the company last year to build our platform. We started looking at this. Let's step back and look at what these peptides mean. So now flashback to the chemistry class. Peptides are small proteins. The cutoff varies by application. The FDA says 40 amino acids or somewhat more if you're looking at insulin-like compounds. For us, we don't have such a strict boundary. It can be as high as 70 or 100, or it can be as small as 10 or fewer. There are 20 standard amino acids. There are over 30,000 non-standard amino acids. These can be linear, they can be branched, they can be cyclic. And the point, though, is that because they are modular, they can be designed to bond to specific molecular targets to mediate protein-protein interactions, have high selectivity, and have different sorts of physical, chemical, pharmacological properties. At the right, we just have a couple examples. Oxytocin is a hormone associated with giving birth. Insulin, we all know. And these give you an idea of their complexity. The targets are even much bigger. Oxytocin has 9 amino acids. Human insulin is around 60 or 70. And these get complicated. But these are the things we're trying to optimize with our AI algorithms. And it is a complicated problem. To give you an idea of that complexity, the standard way of doing this sort of drug design now is what what's called high throughput screening. And these high throughput screening libraries typically have on the order of 1 trillion compounds, or 10 to the 12th. If we look only at linear peptides up to 30 standard amino acids, only from those initial 20, the number of combinations is 10 to the 39th. Now, the ratio of 10 to the 12th to 10 to the 39th is analogous to a drop of water compared to the aggregate volume of the oceans in the world. And so what that means is when we do high throughput screening and we start with 10 to the 12th compounds, the odds of finding the ones we really want in that random library of 10 to the 12th are infinitesimal. That's why we need to go to an AI-based method. When we look at non-standard amino acids, it very quickly goes much higher. So length up to 50 with those non-standards, now it's 10 to the 181. That's a big number. It's comparable to what we've seen before where AI has had success. Things that we did, and I spoke previously at this conference about, with no limit Texas Hold'em poker. That was 10 to the 161. And what others did with AlphaGo. To put that in perspective, the number of protons in the observable universe is estimated about 10 to the 80th. So these numbers are around 10 to the 80th squared. And that's what we need to do to be smarter about this than random library search. How we're doing this is bringing together what this conference is all about. Novel algorithms, both deep generative models, predictive models, they're discriminative. Putting those on to purpose-built supercomputing, where we have designed our system to be specific to peptide drug design, including deep learning, traditional machine learning, and molecular simulation, along with serving very large-scale knowledge graphs and being able to do queries across them. Being able to bring in advanced new technologies like Cerebrus or others. And then the third track here is peptide research and development, where we're building a custom peptide synthesizer to bring down the cost and the time of chemically synthesizing new peptides to collect very rich data, because we need that to complement the compute. So together, these are what lead us to where we are today in terms of being able to discover things. Now, a very quick word about our compute. We built our own on-prem system. It builds on concepts we actually learned from systems we designed previously, like Bridges 2, Neocortex, the Cerebrus system that's running now at PSC, for those with like academic access, HubMap, the Human Biomolecular Atlas on single-cell data. All of those informed what we've done now, and we've gone beyond that to build a system specific to peptide design, and it will scale as we need. We're hybrid cloud. We can burst to public clouds if we need additional compute. We have not needed that yet. And we may serve some software platforms there as well. So the way this works is that we curated and harmonized initially about 70 very large public and proprietary data sets. Since then, we've added quite a few more. These are mainly cheminformatic, bioinformatic, very diverse, very heterogeneous. But bringing those together so we do queries across them, assess data quality, pull out what we need to then do our training. From that, we specify the target and the biology we would like around that target. Do you want an agonist, an antagonist, or so on? And then we have a whole toolbox of deep learning algorithms we have developed. We have deep generative models, and their purpose is to, given a target, we'll show you what that means, to be able to predict a molecule that will bind to that target and have certain properties. We also have a number of individual predictive models that can predict additional properties like solubility, stability, membrane permeability, and so on. We also look at new IP because we're not interested in just rediscovering things that have already been considered. And we're interested in being able to predict synthesizability and manufacturability, because it's also pointless to spend our time on compounds that we cannot easily or cost effectively make. Those models produce a list of novel ranked peptide lead candidates. So these are things we might think about taking eventually for preclinical development and further into the clinic at the right time. We synthesize and validate those. So these give us a real wet lab perspective on whether or not this works. Everything feeds back into our knowledge graph, into our databases. So we iteratively improve as well. The point of the synthesis and validation is both to give us feedback on our models and allow improvement. It also means that we don't need the same level of explainability as we would in certain other medical fields, because all of this is being tested. In a sense, it's not different than if a medicinal chemist came up with a compound based on their intuition and training. We validate those the same way. So let's see how this works. This was a pilot study we did. It is also around antimicrobial peptides, and we're moving beyond that now. Now all of our disease areas are higher value diseases. But this was to test, can we do in the computer what Jonathan, as an expert biochemist, did in traditional ways. The keys here are from starting with data through developing the encoder, scoring it, and wet lab synthesis and assays. End to end was about 7 weeks. The plot on the right shows us the activity of these compounds. And this one, we're looking for potency. So lower is better. That means highly potent, fewer micrograms per milliliter. And the dark bars that go all the way across the plot mean that these compounds are active across all 12 of those tests or assays. So those are really good. And the takeaways are, first of all, this took the process from 5 years to 7 weeks. There's a 25-fold hit rate improvement. So 20% of these predicted compounds show high potency versus industry average of 0.8%. And we're seeing this great potency. So overall, that's a win. Now, looking deeper into what those compounds look like, we took the top 10. Again, these are lab validated, these are not predicted, as the most potent. And we plotted them. This is a UMAP, essentially dimensionality reduction. So each of these clusters denotes a group of peptides that are similar in certain ways, represented in the latent feature space. Overall, there are about 20,000 peptides in this plot. And we see the number of clusters. Those in gray ovals are from known design space. Those outside the ovals are novel. So that's where new discoveries can be made. These two compounds, numbers 2 and 4, are in clusters numbers 13 and 18. So what that told us is this model is predicting compounds outside the training set. They have high potency and they're completely novel. So that was great. We moved on. As I said, anti-infectives are important, but we need to move to more pressing disease areas. So what we're looking at now in our internal discovery pipeline are a rare genetic disease and 2 targets in oncology, 2 different targets. And we're pushing those in our discovery programs. The way this works currently, we also push way far beyond the variational autoencoder that was in the prior model. Now we can predict, given either a target structure, and by that I mean these 3D structures like everyone has seen coming from AlphaFold, or just a target sequence, which is just a sequence of amino acids. Those can be processed by a generative model, and in addition by our peptide scientists, to produce about 1 million, typically, sequences that are predicted to have high binding. And those are ranked by essentially branding probability. Those are then run through a set of predictive property models where we look at things like solubility, manufacturability, and other parameters that say whether or not these actually make sense. We typically look at those and we cluster them, and we look at representatives using molecular dynamics and by docking. The point of that is to be able to, especially for novel peptides that have never been seen before, to understand the biophysics. What are they really doing at the molecular level with those targets? From that, we get about 100 compounds. It could be 100, it could be 300, but on that order. And those are then synthesized. The sequences look like these. There's a high diversity. And they're synthesized, they're assayed. And from there, we measure things like the dose response curves, the activities. And we can either proceed to preclinical, or we could go through the loop again and return to a second cycle where we get better. The main difference between what we are doing and traditional drug discovery is that traditionally, properties are layered on. So one starts by finding a hit, and then one layers on pharmacological properties, things like safety, toxicity. And that's where things break down, because the initial hits often cannot be optimized in these other ways. And that results in essentially a dead end. But by putting all of the properties up front and then iteratively improving them, we don't have these dead ends. What we see here is what's called a G protein coupled receptor, or a GPCR. This is for that rare genetic disease. And we're comparing our designed peptide in blue to a marketed drug in red. And the key here is this model was trained only on the sequence. It was given only the target sequence of this very large set of 7 helices. It's the GPCR. And there's several outcomes. One is the designed peptides bind to the right target. They bind to the correct active site, as we can see in the figure on the right. And then through further simulation, our computational biology team solved the inactive structure of this GPCR, which has not been known. And we find that when bound, these peptides cause the right conformational changes down in the tail of the GPCR that is in the side of the cell, and thereby activate the right antagonist properties, so they have the right pharmacology. And it means these models are actually working for what we need to develop new drugs. That's where we are today. In summary, what we're doing is applying deep learning, especially generative models and also predictive models, on a set of proprietary representations that we've developed to be able to predict peptide drugs and drug candidates for, right now, arbitrary molecular targets, and to be able to do this in a cost-effective, rapid way. We're moving forward on all of our discovery programs. We are in discussion on possible partner programs and open to others. I'm up to time, so I would like to leave it here for any questions.